Hey everyone, this is your host, Dr. Marie McNeely, and this is episode 624 of People Behind the Science. This week, we are excited to be rebroadcasting our interview with Dr. James Pipas, originally published on February 18th of 2015. Listeners, if you haven't met James yet, he is an accomplished molecular virologist. He is investigating how viruses work, how they interact with us, and where they come from. His research has spanned a variety of different viruses, including viruses that cause cancer in humans. James is also conducting innovative work to develop molecular and computational tools to explore viral diversity, conduct surveillance on viruses in nature, and to understand why viruses may spread. So listeners, we hope you enjoy this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am thrilled to be here today with Dr. Jim Pipas. So welcome to our show, Jim. How are you?
Doing very well. How are you today?
I'm excellent and delighted to chat with you today. Now, listeners, Jim is the Herbert W. and Grace Boyer Chair in Molecular Biology and Professor of Biological Sciences at the University of Pittsburgh. He received his PhD in Molecular Biophysics from Florida State University, and he completed his postdoctoral training at Baylor College and the Johns Hopkins School of Medicine. Now, Jim has been a faculty member in the Department of Biological Sciences at the University of Pittsburgh now for over 30 years. And he is here with us today to tell us all about his own journey through life and science. So, Jim, we are excited to chat with you today and get to know you, both as a scientist, but also get to know you personally. So can you take it away here in the beginning and tell us what you like to do when you're not doing science?
Well, okay, great. Well, I have two passions aside from science. First, I'm an entertainer.
All right.
Right. So I've been an entertainer pretty much all my life. My brother, Mark Pipas, is an actual professional entertainer. And I've worked with him for a long time, in both in live performance and video performance and other formats. And so I like to compose music. I like to sing music. I like to produce shows. And we go all around the world doing this. So this is a major part of my life, is the entertainment world.
Oh, wow. That's wonderful.
And then the second thing I like to do is I like to go to remote places like forests or mountains or deserts where there aren't any people and just explore them. So I'm a bit of a naturalist that way. And so those are the two things I spend most of my time doing when I'm not doing science.
Well, that is incredible. I love that juxtaposition, kind of the hustle and bustle of the entertainment world and then just going away to a remote place to escape from it all. That's incredible.
Yeah, they're totally different things.
Absolutely. So what is your favorite form of entertainment? You said you compose music, you're a bit of a singer. What would you say is your specialty?
And I don't think we have a specialty so much. I think if I talk about enjoyment, I really like the thrill of live performance. And I like the shows that we do that mix different formats. They have video, they have theatrics, they have real music. You know, that's kind of exciting. And I like the whole part of producing and composing those kinds of shows.
Very cool. Well, Jim, wonderful to hear about how you spend your time when you're not doing science, exploring the world of entertainment, getting out there, enjoying nature. But now we are going to dive into that scientific side. So can you tell us next a little bit more about how you describe what you do to someone who might not be familiar with science?
I guess the way I'd say it is that our laboratory studies viruses. And we really study viruses for a couple of reasons. One is we want to know how viruses work and how they interact with us and where they come from. And then the second thing we use viruses for is to understand how our own biology works. We use them as tools to study ourselves. So, for example, we study viruses that cause cancer. And we want to understand, by studying these viruses, we learn both about the viruses and how they contribute to cancer, but also we learn about our own bodies and how they regulate cell proliferation and how they prevent cancer.
Very interesting. Well, I think this is a really hot topic right now. I mean, talking about viruses in light of what's going on with Ebola, I'm sure you're getting tons of questions flooding in.
Oh, yes.
Yeah. Certainly keeping you busy. Well, and I know scientists tend to have packed calendars, and it sounds like you especially kind of coordinating performances, practices, getting out, doing some traveling, and then also getting all your science done. That's a lot on your plate. So how do you keep yourself motivated, Jim? Do you have a particular favorite quote or maybe a saying that you love to keep in your mind as you go through your day?
I do have a quote that I think I tell myself a lot, and that motivational quote is, spend as much time as you can with highly motivated individuals.
I like that quote. I think that's fantastic. I've heard the one about, you're sort of the sum of the 5 people that you spend the most time around with. But I think this is really key, spending your time with motivated people. It's almost contagious. It keeps you going as well.
That's right. It's really exciting to be around people that are excited. And so I think that's the best way to help yourself get motivated.
Absolutely. I think it helps being surrounded by scientists who are often very motivated, highly driven people. So science is a great place to be for that aspect. Now, Jim, can you take us back and tell us a little bit more about your journey through science? In particular, let's start off with chatting about some of these people along your journey who might have inspired you or even served as role models for you.
Yeah, the role model thing is a little tough for me because the way I look at life is I don't have an individual role model, I'd say, or even a name I could give you. But what I do is I look at all people, everybody I interact with, and everybody has unique characteristics and they have unique approaches to people and problems and getting things done. And so for role models, I use everybody. I try to look at individuals and how they respond to situations, and I try to incorporate the responses that I like, and I try to avoid the ones I don't. I guess everybody's my role model.
Gotcha. You're building a bit of a Franken role model, pulling things from all different people. I like it.
Yeah, right.
Well, Jim, that is fantastic to hear about how you get inspiration from a lot of the people in your life. Are there certain traits that you've been thinking about recently or seeing in other people recently that you'd really like to incorporate in what you do and how you live?
Well, I think one of the most important things is a positive attitude towards getting things accomplished. It's easy to be negative. It's easy to figure out or come up with reasons you can't do something. But I like people that have a positive attitude and are going to say, we're at this point now. We want to move to this other point. How do we get there? And let's do it.
Absolutely. I think you're building a bit of a theme here, kind of keeping that positive attitude, keeping that forward momentum, that motivation going forward. Definitely important in the career of a scientist and just getting things done in life, I think, in general. So great to hear about some of these people who might have inspired you, some of these traits that you might have pulled from them as you go through your journey. Now, can you take us back to the beginning, Jim? I'm curious to hear about how you got started in science, especially with all these different passions that you have in your life. What really got you glued to science?
Now, that's a good question. So, way back when I got an undergraduate master's degree from the University of Southern Mississippi in chemistry with a double major in microbiology. And the reason was I wanted to be a molecular biologist, and there was no molecular biology at that school at that time. But part of me always wanted to be a scientist, even as a very young kid. The other parts of me wanted to be an explorer or a musician, but that's a different thing.
Gotcha.
Right.
You're still able to do those things, right?
No, I think, it's worked out well. So I had a lab in my basement, when I was growing up, and I read lots of science. And my grandmother, Louisa Moore, she was a teacher and a librarian. So if ever I expressed an interest in anything, if I was interested in dinosaurs or the weather, a book would suddenly appear on that topic. And that really had a major influence on me because no matter what I showed curiosity in, I got support from the family. And so that stimulated my science a whole lot. And I thought when I went to college, that's what I wanted to do. And then when I got to college, maybe I was a little rebellious and the introductory science courses bored me stiff.
I'm sure you're not alone on that one.
Started drifting perhaps. But then it happened. I took Dr. Biedenbaugh's organic chemistry course. And what a difference a great educator makes. He induced me to realize I really did love science. And subsequently, I really, really liked the upper level science courses, physical chemistry, biochemistry. And then I took a course in computer programming way back in 1970, and boy, did I like that. So that really did it. And then I got incredibly lucky to be accepted into the Molecular Biophysics graduate program at Florida State University. And I don't know if you know about this program. This was a program envisioned and created by a National Academy member named Michael Kasha.
Interesting. I didn't know that.
Yeah, he was way ahead of his time. This guy was not only a quantum chemist, but he designed guitars, and Kasha guitars are still a classic.
Really? So he had the same passions, music and science.
Yeah, maybe at a little different level than me, but yes, he did. But this program in molecular biophysics only took 4 graduate students each year. And the idea was to train physicists to become chemists and biologists to be exposed to and maybe converted to physical biochemistry. And that is something that I think is a great need today, that interdisciplinary approach to education.
Certainly.
Right. And so I worked on a PhD there on bacteriophage called P22 in the lab of Bob Reeves. But during then, I had another great educational experience. I met a fellow graduate student who liked to go mountain climbing with me, Jim McMahon. And together we wrote one of the first computer programs ever applied to biology, way back and published in 1975. And so all those things really stimulated me into becoming a scientist. And my postdoctoral training was just with super people. You know, when I was coming out of graduate school, I really targeted 2 labs to go to, Paul Berg at Stanford and Dan Nathans at Johns Hopkins. And Dan Nathans, he told me that it's going to take a year to get into his lab.
Oh, wow.
And Paul Berg said it's impossible to get into his lab. But he recommended a recent postdoc from his lab, John Wilson, who had just become an assistant professor at Baylor College of Medicine. And so, wow, did it work out well for me. I went to work with John. He turned out to be a great mentor, a super scientist, and a friend, and that friendship continues today. And then I moved to Nathan's lab a year later at Johns Hopkins, and that was just incredibly exciting. It was at a time that restriction enzymes had been discovered and cloning was being done. And during my stay there, he actually won the Nobel Prize.
Oh, wow. So what was it like being in the lab when he got that award?
We were all in a good mood.
Right. If the boss is happy, you're happy, right?
That's right. He was a very humble and unassuming man, and that taught me a lot too. You know, that he said, it's really the science that we're here for, not these prizes. And so, yeah, that was a great experience. And then I moved to Pittsburgh, and here I am, a scientist.
Right. Well, Jim, it sounds like you have had just a fantastic journey through science. And you said a couple of things along the way that really caught my attention. You mentioned that you had a lab in the basement as a kid. Can you tell us more about this lab? What was in it? What did you do in this lab?
Yeah, just about everything, mostly chemistry. This was in part of my basement where my mother had the washer and dryer. And bless her heart, more than once she came down to find a frog or a snake sitting on her dryer.
Right.
So this had, you know, it had little centrifuges and little tubes. I really did chemistry. I was into it. And it was a whole little part of the room.
That is amazing. I think a lot of us, when we look back on our childhood, we may not have had a basement laboratory, a whole big space, but, you know, maybe a little corner of our room or a little place in the kitchen we'd like to go to do some of these early quote unquote experiments. And I think it's so important to have that support of your family to foster this early curiosity. So great to hear that you had that support. Your mother was willing to overlook maybe some of the frogs and snakes that appeared in the basement. So
yeah, made for good stories.
That's right. That's right. And I think it's fascinating to hear about the courses that really hooked you. You don't hear too many scientists mention things like organic chemistry or computer programming are the science courses that really got them excited to keep going in science. So very interesting to hear your story.
That really did it, though.
And I think the instructor can make all the difference, right? So if you have like you said, a really inspiring professor, someone teaching you the material who's really excited about it, I think that can make all the difference. So Jim, we've talked about your journey through science, and now you mentioned you're here at the University of Pittsburgh having a great time doing science. Can you tell us about one of these projects that you're working on now that you are just the most excited about?
We're excited about a bunch of them, but I guess the story I'll tell you about today are looking for new viruses. So if you go to a textbook or to the various committees that make these decisions, they'll tell you there's about 4,000 species of viruses that exist on planet Earth. And yet, so we've been doing experiments to go to the different environments around the world and just sampling them and asking how many viruses are there and how different are they from each other. And so what we found from these studies is that the 4,000 viral species that are listed in what's called the reference sequences, these are a tiny, tiny fraction of the number of viruses that are in our world.
So if you had to estimate or project, how many viruses would you guess right now there are out there?
You know, these questions have been asked.
It's the $1 million question, right?
It's really, really difficult to get that. But what we can say is if we just survey nucleic acids that are inside a virus particle in some environmental source, for example, raw sewage, that the number of sequences we get that don't match anything in any database outnumbers those that do by about 100 to 1.
Oh, wow.
And so these are viruses that we know so little about, we can't even match their sequence to anything. And so it tells us that the 4,000 viruses is very small. But not only that, if you go to a virology textbook, you'll find that that textbook describes 40 or 50 viruses. Those are the ones that we really know a lot about and are studied. And so I guess what's exciting to me about this, because I'm a scientist and I like the unknown, is that we don't know diddly about most of the viruses in the viral universe.
Certainly. Well, it sounds like this is a really big sort of new wide open frontier. I can't even imagine sort of 100-fold increase over what's currently out there needs to be described.
Well, we think pretty sure that's the minimum. It's probably much, much more than that. And if you think about it, the numbers, you can actually do the calculation yourself. So we think that almost every species on planet Earth has viruses associated with it. Mm-hmm. And the World Conservation Union tells us there's about 2 million species on planet Earth. So already 2 million is much more than the 4,000.
Right. So where do you start with this, sort of wide open arena? Where do you start in your search for new viruses, Jim?
First of all, we take advantage of the fact that viruses protect their genes inside of a particle called the virion, the virus particle. And so what we've been doing is actually looking at raw sewage initially, because raw sewage contains deposits from humans, contains deposits from animals, and then there are all sorts of creatures that grow in raw sewage. It's its own ecosystem.
You're making it sound very glamorous. Keep going.
Come on. And we can collect raw sewage around the world, right? And
right.
In different environments to ask, how do the viruses in these different environments change? So that's where we start. We start with a sample, but it doesn't have to be raw sewage. Another thing we're looking at is mosquitoes. Just collecting mosquitoes from everywhere, or ticks. Mash them up, isolate the virus particles, and then sequence all those virus particles to get an idea of how much diversity there is out there.
Very interesting. So how are you able to separate the viral genetics from what might be there from other organisms?
That's a really good question. So again, we're taking advantage of the fact that the viruses protect their genes inside of a virion. So if we go to raw sewage, we treat these samples with enzymes that destroy DNA or RNA. And it'll destroy all the RNA, DNA in that sample, except those nucleic acids that are protected by a particle. And so that's the first step, really. We get rid of most of the other nucleic acids biochemically. And then the next step is computation. So we can also subtract these other sequences out computationally. And that's one of the big parts of this project, is developing the computer tools to recognize viral sequences and distinguish them from other things like cellular sequences.
Definitely. Well, it sounds like those computer programming classes are coming in handy.
Well, actually, I have people that know a lot more than I do about that doing that now.
Gotcha. Well, Jim, it sounds like this is just an incredible project that you were working on with a lot of work cut out for you still. So.
Oh, yeah. But it's a lot of fun.
Absolutely. Well, very, very interesting. And I think just a phenomenal new frontier, like I said, to continue exploring these viruses and definitely considering humans. I feel like most of the viruses that we know about are the ones that are big health issues in humans. But thinking about every species out there having its own associated viruses, that just blows my mind in terms of the scope of these questions that you're trying to answer. So very, very interesting. Now, Jim, we've talked about a lot of the great things about your career as a scientist, the work you're doing now, things like that. We haven't yet talked about some of the tough times, some of the struggles. And I know every scientist out there, every person out there can think back to points in their life where they were failing majorly, or maybe just having incredible struggles doing something. So can you think back to one of these tough times and share it with us today? Because I think what you see in science is often the success stories, the result, the end point of the journey. You don't always get to hear these difficult points, these road bumps along the way.
Yes, let's see. A single time?
You can pick your favorite.
It's hard to do. Science is so full of struggle and tough times. And there's so many times that I have made mistakes, or I've made good decisions that just didn't turn out right, or I was just plain wrong about something. It's almost hard for me to focus on one. But what I'd say is that each one of those obstacles is exhilarating. It's exactly what scientists do, right? If it were easy, who would need scientists?
Right.
Right. And so 100 of these things come to my mind. I don't think I could focus on one. I'd say it's important through all that to keep a positive attitude and plot your course forward.
Absolutely.
And I got another quote. Maybe it's not a quote because I'm not sure I can do it exactly. But one thing I think about a whole lot is a statement by Thomas Edison, who said something like, "I have not failed. Rather, I have discovered 10,000 ways that don't work."
I like that. I have not heard that quote before. I think that's a great one.
I hope I assigned it correctly because I think about that one all the time. I especially think of all those times I've had great ideas that I'm sure would solve a problem. But then experimental results just said, "Jim, you are wrong."
Right. Well, I think that's a really important part of science that I think maybe people don't realize, especially when you're young and considering careers in science. There's a lot of exciting elements of discovery, of, making progress. But a lot of science is doing it wrong the first time, the first 10 times, the first 100 times before you get it right. So great point to bring up. Now, can you tell us about one of these struggles, one of these challenges that you faced maybe in the past week?
Well, I guess the one we're thinking about right now is how to look at very rare viral events. So the one I'm thinking about is there's a type of virus called the Khaleesi viruses. And the most famous member is Norwalk virus. This is the one you read about in the newspapers because they have to shut down a cruise ship line.
Gotcha.
Because all the people have, the term is explosive diarrhea.
Mm-hmm. Again, glamour, right?
Very glamour, right? So this virus is a major health problem. It turns out a lot of the foodborne illness around the world is caused by this virus. And we keep getting it because it mutates and changes, right? So our immune system can't recognize the new virus that comes around. But how do you study that? Because if you look at a population of viruses and grow them in bulk in a test tube or a petri dish, then there's millions of virus particles there. And there may only be a few, 1 in a billion, 1 in 100 million that are different. But those are the ones that are going to be the problem. And so the obstacle we've run against is, how do you get that 1 in a billion?
Mm-hmm.
How do you think about that? Do you want to hear the solution we're using?
I would love to hear the solution, or even how you got to it. I'm intrigued by this problem.
Actually, if I have a second, I'll tell you another story about how we got to this solution.
Sure.
It's a lesson in science. All right, I was thinking about this problem. If I have one virus I want, or 100 million other viruses I don't want, how do I capture my one? And one thing I thought about was, in my dreams, I said, okay, what you'd do is you'd set up 1 billion test tubes. And then you could do polymerase chain reaction, PCR reactions, looking for your one virus and find out which test tube out of the billion your virus is in.
Mm-hmm. You need a big lab, but I think you could do it.
Exactly. Or highly motivated individuals, right?
Right.
So obviously this was not practical, right? But I went to a meeting, and it's not even important what the meeting was about, but I was sitting at a table with a young man. His name's Asaf. And we started talking about, what do you do? And I told him, I work on viruses, and here's what I do. When I asked him what he did, and he said, literally, I blow bubbles.
Wow.
And I said, really, how do you make a living blowing bubbles? And he said, well, I can blow bubbles precisely the same size every time, and I can blow billions of them. This technology is called droplet-based microfluidics. And Asaf is a postdoc in the laboratory of David Waits at Harvard University. And the key part of this conversation, when I said, well, what do you do with the bubbles? And he said, well, think of each bubble as a test tube.
Uh-huh.
Then I said,
light bulb moment.
Right. Billions of them. So we've been working with the Waits lab to use this droplet-based microfluidics. And we've now shown that we've solved, I think, the first big hurdle. And that is that we can detect a single virus particle in a single bubble, in a single droplet, and sequence it. And that allows us to see these very rare variants that are present in a viral population. Now, we've still got problems ahead of us, but that's a wall that we've hit, and I hope we've blown through it.
Jim, I think that is a marvelous story of collaboration coming through in the end in science. And I think this happens so frequently. It's these chance conversations that you have with people, whether it's at a conference, at a pub, at a lab meeting, things like that, that just sparks something that you're like, oh my gosh, I can apply this to this problem that I'm having right now and get to a solution. So really, really great story there. And I know we mentioned this was a bit of a struggle, a challenging problem that you face in your own laboratory. We don't just want to talk about the tough times, even though this one seems like it's going to have a happy ending in the end there. But can you tell us next about a success story, one of these moments in science that really meant a lot to you? It can be a big success, like getting a grant or a paper published, or even one of these small victories along the way.
Every day we have the small victories, and that's what keeps us all going. right? So I guess one of the success stories that I tell you about right now, it has to do with these viruses we work on that cause cancer. So we've been curious, how do these viruses cause cancer? And the whole focus in cancer has been on genetics. The field thinks that mutations in specific genes drive tumorigenesis, drive tumor formation. And that what viruses do is they encode for proteins that bypass the need for some of these mutations by directly altering the cell. And so we've been working on this particular viral protein encoded by a member of the polyomioviruses. These are cancer-causing viruses.
Gotcha.
And what we've been able to show is that it's not just the genetics. But it's rather the type of cell that you produce this protein in that results in either tumorigenesis or no tumorigenesis.
Interesting.
Yeah, so if we take this one protein and put it into the intestine of a mouse by various tricks we can use, then that mouse will have abnormal cell proliferation but no tumors. But if we put it into the stem cell compartment of the intestine, now you get full-blown tumors. And so you see the genetics is the same in both of these cases, but what's different is where the genetic alteration occurred. And we think this is actually going to be very important in cancer research, to focus on what cell initiated the tumorigenesis and how that governs the properties of the eventual tumor.
Well, congratulations on this success story. This sounds like a really exciting direction for your research to take. And like you said, the implications in cancer, really, really fascinating to start to think about, where those tumors are starting, where those viruses are taking action and why they're not taking action in other cells. So fascinating work. Now, I know researchers keep so busy doing the work that they do. And you also mentioned you have all these things that you like to do outside the world of science. I think it's hard to find time to sit back, relax, and sometimes enjoy a good book. But I've gotten some great book recommendations from Scientist Jim. So I'm going to ask you for one today. Can you tell myself and our listeners out there what book you've read and just really enjoyed that you think we all should check out?
All right. I guess the one I would pick is A Hitchhiker's Guide to the Galaxy by Douglas Adams.
Wonderful recommendation. Why did you like this book?
Well, I especially like the part on the Hitchhiker's Guide that says, "Don't panic."
Right. I feel like we all need a don't panic button. Everyone talks about the panic button, but...
That's right. I think the don't panic is really good advice. But also, I think the book actually is a great commentary on the way the world works and on human nature. And it's a simple read.
Absolutely. Wonderful recommendation. I really enjoyed that whole series myself as well. So I will put that out there for our listeners to check out. And I think we've chatted about a lot of different things today, and we've talked about different opportunities that scientists have, things that they like about their career, things that they don't like about their career. And I think one of the things that scientists love are some of the opportunities that you get for travel. And I think these opportunities come in a number of different forms, sometimes for conferences, sometimes maybe to collect sewage somewhere. But can you tell us about your favorite place that you've traveled for Science Jim, and why was it so memorable?
Well, I've been really fortunate to travel a lot of places, and that's one of the things I recommend to students, by the way, is travel as much as you can. That is a great education. So I'm not sure I have a favorite place, because every place has a place to offer, but probably my most memorable trip was a trip that was called Around the World for Science and Music. And this was a 4.5-month around the world trip that my brother and I took in 1989.
Oh, wow.
And I was giving scientific lectures and he was giving concerts, and then sometimes we worked together on the concerts. 1989 turned out to be a really pivotal year in world history, so it was a great time to take this around the world trip. We started with a multi-country lecture concert tour in Europe, and then we entered the Soviet Union from Finland and through Estonia and Lithuania right at the times when these Baltic states were rebelling against the Soviet Union. And went up to St. Petersburg and down to Moscow, where our hostess was the daughter of the editor of Pravda.
Oh, wow. And
so really got to see the Soviet Union from a unique vantage point. And then we took the Trans-Siberian Railway all across Siberia, getting off in Novosibirsk and Irkutsk, and arriving in Beijing, where I gave some lectures, and over to Xi'an and Hualien, Hangzhou, and Shanghai and Hong Kong, and then down to Taipei and Bangkok and Singapore and Manila and Indonesia, before ending up in Australia and New Zealand. So, and all the way along, I met scientists and musicians that, we still stay in contact with. So that's got to be the most memorable trip.
Jim, this just sounds like an absolutely life-changing experience. So I have to ask, how did you swing that? Was this on a sabbatical, or how did you get this lined up?
Yeah, it was on a sabbatical. So it was on a sabbatical. The first thing that happened was I received some invitations to give lectures, and they were in Europe and Russia, right? So part way around the world. Mm-hmm. And then Mark, my brother, just at the same time got some concert offers. And I started thinking that when I was young, I watched a movie called Around the World in 80 Days.
Right.
There's been a remake of it lately, but this movie made me think, if I'm already in Russia, why don't I just come home the other way and go around the world? And when we decided to do that, we started getting additional invitations for lectures and collaborations and concerts. It just sort of built itself.
Oh, wow. This sounds amazing. So is there a particular place that sticks out in your mind where something happened that was maybe contrary to what you expected, or a place that was maybe more exhilarating, more beautiful than you could ever imagined it would have been?
I think probably Siberia. A lot of biology that's unique there, untapped, untouched forests and mountains, and very remote. So it's got the biology, it's got the remoteness, a lot of the things I like.
Absolutely. Well, this sounds like an incredible experience. And I think being able to take these long sabbaticals, being able to see the world is such an important part of science. You know, getting out there, forming collaborations, even just getting inspiration from being out in nature, being in different places, different cities, things like that. So wonderful to hear about this incredible trip that you were able to take around the whole world. Now, I think in addition to some of these travel opportunities you get as a scientist, and I think as you progress in your career, the travel becomes more and more, and you get to make these connections, make these collaborations with people, like you said, that you continue to have contact with over the years. And when you think back over these groups of people that you worked with, these people that you spent time with, I think it's funny to look at the different personalities of groups that arise. And I think with all these brilliant people working closely together, you get some quirky traditions that spring up, or even just some funny memories that you can share. So can you tell us about one of these traditions, or even one of these memories today?
So I'm probably not very good at that.
Nothing quirky happened going around the world?
Oh, no, nothing I can talk about.
Oh, gotcha.
No, I don't know about quirks or strange traditions, but all the labs I've been associated with, they were populated with the scientists and staff who like to work hard and they like to have fun. And all of them worked really hard, and all of them had a lot of fun. So especially back in Dan Nathan's lab, we had lots of interactions among all of us that were working there, the graduate students and the postdocs. Mostly where we would take off and do something we just weren't good at. You know, we were so good at being scientists that we like to explore things that we weren't good at, whether it was whitewater canoeing or kayaking or just playing tricks on each other.
Gotcha.
Right. And so I don't know. I don't have any traditions or any trick I could talk about, but it's great being a scientist and working with these people.
Certainly. And I think that's one of those things where there's some stereotypes out there about how scientists are very serious all the time, very intense sort of people. But so many of the scientists I've met have this work hard, play hard mentality where they love to have fun. They like to incorporate fun into the lab, into their work. So is this one of those things where when you're doing your music performances, even, is this something that you bring your lab to, or you invite them to as well?
Actually, the entertainment thing is pretty separate from the science.
All right.
Although we do get hired to play scientific conferences, and we've done that. And we have science songs because, you know, as a composer, any composer writes about what they know. And so I live my world in the world of nanometers.
Gotcha.
And so I've written several songs. You know, one of my favorites is co-composed with my brother, and it's about viruses. It's the SV40 Laboratory Undesirous Adenovirus Polyoma Take Me Home Blues.
Oh my gosh, that sounds incredible. Is there somewhere online where our listeners can hear this song?
You can actually get it on iTunes.
Okay, excellent.
And another one of my favorites, I Got My Ions, You.
I am a sucker for puns and wordplay, so I'm really loving these song titles. I'm going to have to check them out. I'll find you on iTunes out there. That's wonderful.
Well, if you like wordplay, you'll probably like our band.
Fantastic. I will have to check you guys out. We'll send a link out there in our newsletter. So if anyone wants to sign up to get some bonus material, definitely check out our newsletter. We'll put these little links to the songs out there.
All right.
Wonderful. Now, Jim, we've talked about some incredible things today. And I think one of the conversations that I love to have with scientists, this is one of my favorite questions to ask, is not only to talk about the research that you're doing right now at this moment that you're excited about, but some of these big questions that are maybe mulling around in your brain, things that you would love to answer, but there are things standing in your way. And oftentimes it's funding or staff or technology or even feasibility that keeps scientists from answering these big questions. So can you tell us today, if we took away all of these restrictions, what is one question that you are just dying to know the answer to?
Well, I've got one.
Only one?
Well, one approach, I think, that can move science forward.
Sure.
I told you that there's lots of viruses we don't understand, right? We don't even know about these viruses. Mm-hmm. And each of those viruses has genes, and some of those genes are just involved in replicating the virus. But it turns out many viral genes are just there to alter the cell's biology. And we've used viruses, when I say we, I mean the whole field, has used viruses to discover how the cell works. Well, if there's thousands and thousands of viruses that are not characterized, then each of these encode proteins that we could use to study the cell. And we call all the proteins encoded by all viruses, we call this the pan-viral proteome. All proteins encoded by all viruses. And so now, with this microfluidic platform, more powerful computation, better proteomic platforms and sequencing platforms, what I'd like to do, and what we're trying to build a program to get funded to do, is to study all of these proteins at once. That is, develop high-throughput methods where we, instead of taking 20 years to characterize one virus, we could characterize multiple viral proteins anyway in just a couple of years.
Oh,
and
wow.
so I think this is achievable. I think the technologies there is a matter of bringing all the different technologies together and doing it. And that's one of the ways I'm spending my time.
Well, Jim, this sounds like an incredible approach to develop and really an incredible opportunity being able to explore this. I love the name of it, the pan-viral proteome, and really figure out what's out there in these viruses that we might be able to use for science, for medicine, all these important applications. So really great question for us to mull over, myself and our listeners out there. I love thinking about these big questions. So we've had a lot of great conversations today. And I think in addition to some of these big questions that you talk about with other scientists, with your lab, things like that, I think you get a lot of great advice. This may come from mentors, this may come from peers, even people many years ago, advice, whether you've taken it or not. Can you share with us today, Jim, what you would consider the best piece of advice that you've ever received?
Yes, this came from a football coach. Stay low and bend your knees.
And how does this apply to your life today?
It applies because when you walk into a laboratory or any part of life, you really don't know what's going to hit you. And so you want to be low and have your knees bent to be ready to respond to anything.
Brace yourself for impact at any moment, right?
That is correct, because you never know what's going to come and you want to be ready. And if you are, then you can actually take advantage of that surprise.
Fantastic advice, Jim. Now, is there any other last piece of advice or maybe a last piece of inspiration that you want to leave our listeners with today, coming from you personally now?
I'd just say define your dreams, create a plan to achieve your dreams, perform frequent reality checks, have fun, and go for it.
I love it. Sounds like a checklist there. I'm going to write those down and check them off as I go.
All right.
Fantastic. Well, Jim, if our listeners want to learn more about you or your research, where should they go?
I think they should go to the Department of Biological Sciences, University of Pittsburgh website, and all my contact information, my laboratory information is there.
Well, listeners, definitely check out that website. Get in touch with Jim if you have any particular questions. And Jim, thank you so much for joining us today and sharing a part of your story.
Oh, thank you for giving me the chance.
Absolutely, Jim. This was a pleasure. And thank you to our listeners out there. Join us again next time for another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.